The common fruit fly provides a powerful new empirical tool. One of the most important empirical tools used by Morgan and his colleagues was their choice of an ideal model organism to study and experimentally manipulate in the lab, the tiny fruit fly, known as Drosophila melanogaster. With an incredibly short generation time of about 2 weeks, a small set of only 4 pairs of chromosomes, the ability to thrive in laboratory settings, and a broad range of visible phenotypes associated with specific alleles, Drosophila has turned out to be one of the most informative multicellular organism models for generations of biologists. This continues to be true today. Morgan's intrepid group of laboratory explorers made an extraordinary number of key discoveries that extended Mendelian genetics and linked hereditary factors to the formerly hidden inner workings of cells. These new principles and experimental approaches included, but were not limited to, the following. 1. Mendel's hereditary factors, which are now commonly referred to as genes, are physically linked to chromosomes. Most chromosomes contain hundreds or thousands of gene factors called linkage groups. 2. Only when factors are located on separate linkage groups, that is, when they are located on separate chromosomes, do they assort independently. Mendel's 7 factors in pea plants were located on 7 different linkage groups. 3. Most pairs of chromosomes, called homologous pairs, contain the same set of genes in the same locations. 4. Genes often have 2 or more distinct versions or alleles, which can determine different visible characteristics or phenotypes. 5. The pairs of homologous chromosomes may carry 2 copies of the same allele, in which case the individual has a homozygous genotype. Alternatively, they can carry 2 different alleles, in which case the individual is said to have a heterozygous genotype. 6. Mendel only discovered one type of dominance relationship, in which the dominant allele, when present, will mask the phenotype of the other allele, which is said to be recessive. 7. Although some pairs of alleles obey Mendel's law of dominance, others behave differently. Sometimes the phenotype is intermediate, which is called incomplete dominance, and sometimes both alleles produce different visible characteristics, which is called codominance. Human blood types provide an excellent textbook example of the relationship between codominant and recessive alleles. 8. Sometimes one gene produces a visible phenotype and masks the phenotype of another gene. This is called epistasis. 9. Sometimes one gene is responsible for several phenotypes and is said to be pleiotropic. 10. Sometimes multiple genes contribute to the same visible characteristic in an additive fashion. This is one type of quantitative inheritance. 11. Most of the chromosomes are in homologous pairs known as autosomes and carry the same linkage groups of genes.